In this session, we'll see the histology of nervous tissue. You can see here in this diagram, we can clearly make out here this is the axon and this blue color one is the myelin sheath, and here you can see the connective tissue covering the axon and myelin sheath. Single axon and myelin sheath is the endoneurium, and like this a bundle of axons together form a fascicle that will be surrounded by a thicker connective tissue layer called perineurium. And here you can see the entire or the multiple fasciculi are surrounded by the outermost connective tissue covering called as epineurium. So you can see three coverings from outside inwards. If you see, this is the epineurium which covers the entire nerve, and you can see the perineurium which covers the each fasciculi, and you can see the endoneurium which covers individual axon. So peripheral nerves, they are arranged in the form of bundles. Each nerve fiber with its Schwann cell and basal lamina is surrounded by a layer of connective tissue called endoneurium. So it consists of type 2 collagen fibers, fibroblasts, and then within that Schwann cells and endothelial cells and macrophages. Then nerve fibers together form bundles or fasciculi surrounded by perineurium. Perineurium is made up of layers of flattened cells joined by tight junctions. So this form the nerve-blood barrier separated by layers of collagen fibers. So layers of flattened cells separated by layers of collagen fibers. Okay. So these layers of flattened cells which are joined by tight junctions, they act as nerve-blood barrier. Many fasciculi together form a nerve surrounded by epineurium. Epineurium has fibroblasts, collagen fibers and adipose tissue. So this adipose tissue will help in cushioning effect. So in bedridden patients, if this adipose tissue is lost, then there will be pressure effects on the nerves. So nuclei seen in section of the nerve fiber is the Schwann cell and fibrocytes. So here, so these nuclei are the nuclei of Schwann cells and fibrocytes. Now you can see here, so this is the individual axon surrounded by the endoneurium. So this is a single fasciculus surrounded by perineurium, and this will be the epineurium surrounding the entire nerve. So this is the diagrammatic representation. Now you can see the low magnification of a peripheral nerve slide. You can see this is the epineurium. You can see each nerve bundle surrounded by the perineurium. And as this is low magnification, we are unable to see the individual axons. Let us see the higher magnification. You can see here, this is the higher magnification. So this pink color ones are the axons, and the surrounding one will be the endoneurium. So if you see the identification points, each nerve fiber is covered by endoneurium. Each bundle of nerve fiber is covered by perineurium. Each nerve is covered by epineurium. So cut section of axon with myelin sheath and Schwann cell is shown in this diagram. You can see here, this is the axon. You can see the nucleus of Schwann cell, and you can see this is the myelin sheath, and you can see this is the endoneurium. Okay, so whenever peripheral nerve slide is given, at least 2 out of these 4 points should be there to identify the slide in the spotting. Then what is the difference between a peripheral nerve and a optic nerve? Because this optic nerve develops from or develops as an outgrowth of brain. So here this will be myelinated by oligodendrocytes. The first difference, if you see the peripheral nerves, they will be myelinated by Schwann cells. Here this optic nerve is myelinated by oligodendrocytes. And as we have discussed earlier, the peripheral nerve was covered by epineurium, perineurium and endoneurium. And here if you see, the optic nerve is covered by meninges. So it is made up of bundles of myelinated axons which are arising from the ganglion cells of retina. So myelin sheath in optic nerve is formed by oligodendrocytes, as I said earlier. And at the center of the optic nerve, you can see the central retinal vessels. I'll show you in the next diagram. So the pia mater which surrounds the optic nerve, that will send septa which separate fiber bundles in optic nerve. I'll show you in the next diagram. You can see here, this is the dura mater, this is the arachnoid mater, and this is the pia mater. You can see this pial septa coming into the substance of the optic nerve, separating the fiber bundles. So usually this should be the perineurium if you see in the peripheral nerve, but here it is the pial septa. And in the center you can see clearly the central retinal artery and vein. So you can see here, this is a photomicrograph where you can see this will be the, these are the outer meninges, and this will be the blood vessel. Here you can see the pia mater sending pial septa, separating the fiber bundles, and here you can see the central retinal artery and vein. So points of my identification will be, it is covered by meninges, central retinal artery and vein will be seen, pial septa divide the nerves into bundle. So if you see the peripheral nerve, outermost covering epineurium, fasciculi covered by perineurium, individual nerve fiber covered by endoneurium. So here outermost covering is by three meninges. The pia mater will send the septa into the substance of the optic nerve to separate the bundles of optic nerve fibers. Then the myelination of the optic nerve fibers is by oligodendrocytes. Then ganglia. Aggregation of the cell bodies of neurons outside central nervous system are called ganglia. Same thing, if these are aggregated within the central nervous system, we call them as nuclei. Basically two types, sensory ganglia and autonomic ganglia. So autonomic ganglia, there will be sympathetic ganglia and parasympathetic ganglia. So let us deal with the sensory ganglia. Examples are dorsal nerve root ganglia of the spinal nerves, and the sensory ganglia associated with fifth nerve, trigeminal, seventh nerve, eighth nerve, ninth nerve, 10th nerve. Then this sensory ganglia will have large unipolar neurons except in vestibulocochlear nerve where there are bipolar neurons. So this sensory ganglia are covered by well-defined connective tissue capsule. These unipolar neurons will have centrally placed nucleus. Neurons are arranged in groups separated by groups of myelinated nerve fibers. See, myelinated nerve fibers. Satellite cells or the capsular cells are prominent and they surround the cell bodies, and these are continuous with the Schwann cells. Outside satellite cells, delicate connective tissue is present surrounding the cell bodies. So this will continue with the endoneurium of the axon. You can see here, these are the groups of large unipolar neurons. You can also see these small nuclei are the satellite nuclei, satellite cell nuclei. And here you can see the groups of unipolar neurons separated by groups of myelinated nerve fibers. So this is a photomicrograph where you can see these are the groups of neurons separated by nerve fibers. Here also you can see the groups of neurons separated by the nerve fibers. Autonomic ganglia. So these are actually the aggregations of postganglionic neurons covered by thin connective tissue capsule. Multipolar neurons with eccentric nucleus. These are not aggregated into groups. These neurons are scattered throughout the ganglia. So nerve fibers in autonomic ganglia are unmyelinated and thinner. So satellite cells are also less defined. Neural substance is better defined in neurons of autonomic ganglia. You can see here small multipolar neurons scattered, separated by scattered unmyelinated nerve fibers and less prominent satellite cells. And the connective tissue capsule is also thin. You can see here thin connective tissue capsule. So you can see here and there the scattered multipolar neurons separated by scattered unmyelinated nerve fibers. So difference between sensory ganglia and autonomic ganglia. So in sensory ganglia, cell bodies are large. Here these are smaller. Unipolar neurons, multipolar neurons. So here exception is vestibulocochlear nerve. So neurons have centrally placed nucleus. Here eccentrically placed nucleus. So cell bodies of the neurons are arranged in groups. These are scattered. Myelinated nerve fibers are arranged in the form of bundles. Unmyelinated nerve fibers scattered. Prominent satellite cells, less prominent satellite cells, well-defined connective tissue capsule, poorly defined or absent connective tissue capsule. Clinical aspects. Regeneration of axons does not occur in central nervous system because myelination is by oligodendrocytes. In multiple sclerosis, myelin formed by oligodendrocytes undergoes degeneration while myelin from Schwann cells is spared. Loss of epidural fat in bedridden patients causes pressure on nerves. Reduced blood supply leads to ischemic neuritis. Then precursors of neural cells will lead to tumors called medulloblastomas. Germanomas, these commonly found in third ventricle. These are the midline tumors. Then from neuroglial cells, there will be astrocytomas, oligodendromas, and tumors from ependymal cells and Schwann cells are less common. Thank you.